Hello, you wonder which lab to join. You probably have Googled how to consider a lab for PhD, and you see a list of things you need to consider. Your own interest, lab funding status, lab publications, lab members, management style, work ethic. So I'm going to focus on these lab funding status because that's describe the projects in the lab. Probably that's the key you need to consider. So from NIH reporter, you see, type in my name, you see this 4R1 grant. So we have multiple sclerosis, Parkinson's disease, multiple sclerosis, and liver. So it's brain and the liver imaging. And we actually have a new grant come in Alzheimer's disease. In my lab, we have Pascal Spinsamilla, associate professor. He has 2R1 grants. So on the heart, study oxygenation, and on the liver. And we also have another associate professor, San Wang. He has a carotid artery imaging grant. So pretty much we cover from the head, neck, heart, liver, almost all the body parts on all kinds of diseases. For full disclosure, we have a Cornell spin-off company, Magic, commercializing QSM. All right. You wonder how to consider a PhD project. So I suggest a principled approach using the NIH grant review criteria. So that's 5 items: significance, investigators, innovation, approach, and environment. You can look it up on the NIH website. So let's go through significance. So your PhD project need to contribute to healthcare. So the 2 aspects need to be relevant to healthcare. So study diseases, right? Need to try to improve healthcare, like any aspects, diagnosis, monitoring, understanding, treatment, and so forth. All right, so we have significant investigators. So you need to join a team of technology and medical experts. So we have leaders in MRI fields, and so we already mentioned me, Pascal, Fan, and we just hired 3 new faculties: Simon Winkler, Stephanie Hector, and Jean Kim. They joined our MR Research Institute. And we have leaders in medicine from radiology, neurology, neurosurgery, pathology, hematology, and cardiology, and so forth. So we have a productive track record working together. So we are doing good on investigating. Now, innovation. So your PhD project needs to be innovative, need to be able to contribute something. So here we use computation, physics, biology to attack any diseases in any organs almost. Inventing new imaging algorithm for data acquisition or data processing, or try to discover something new in biomedicine. All right. So I'm going to give you some background regarding 2 major technologies invented in the lab. So one is called quantitative susceptibility mapping QSM, which is to study tissue magnetism. Another is quantitative transport mapping, which is study tissue transport. So tissue magnetism. So not the biometals. Here you see iron is in every part of the cell, which is like a chemical plant. Iron is a commonly used catalyst for all chemical plants and here for all biochemistries. So and iron is highly paramagnetic, and so let's review some magnetism physics. Iron carries a magnetic, not a charge, but called dipoles because we don't have magnetic charge. And then you have the fields, it's called dipole fields, and has special magic angle 0. When we do imaging in a voxel, because the field variation in the voxel, then you have destructive interference, so that's so-called T2 star signal hypointensity or signal decay. Otherwise, you have a phase. So this is the basic magnetism physics, and when we do imaging, so we can map the fields, which is a convolution of double kernel with the susceptibility distribution. So susceptibility carries describe the magnetic moments of tissue. So we have this double kernel convolution, and so it's just AX equal to B, right? So then you solve the inverse problem in this manner. Using numerical optimization or the classical machine learning or latest deep learning. Here, regularization is needed because the inversion is ear post. So here, just showing some example of images. The magnetic images we acquired, and usually we also acquire phase because our data acquisition is always complex. Then we clean it up, estimated fields, and then we do inversion to the susceptibility. So you see the bright signals, the deoxyheme ion in the veins. And then you see the bright signals in the basal ganglia. That's ferritins involved in neurotransmitter and neural diseases. Okay, so then you see the negative classifications. Right. That's the basic question. Next phenomenon we studied is transport. So this circulation is vital to deliver nutrients, oxygens to tissue. And so if you look at detailed modeling, so we see blood flow, there's a perfusion, which is really advection, and then diffusion, right? So that's underlying physics, that's called transport. But we are doing using this Caddy's equation, which ignores the spatial derivatives using a global arterial input function. That's purely wrong. It's crazy stuff in medicine. We need really bring precision rigor to medicine in here. So here it's just transport equation. The variation in time is equal. It's related to gradient of variation in space. So that's the continuity equation. And this is linear in velocity we want to map. And so that's a simple AX equal B problem, inversion, right? So we can solve it similarly, but of course the underlying is a little bit different. So we showing here an example, the kidney. We can build the vascular tree structures, and we already showed we can generate the velocity maps. So we actually did a nice validation. This is ever the first time we used the computational fluid dynamics calculation to generate the velocities, the flow fields, and further simulate the 4D imaging, right? And then we do the processing using our QTM approach, and we also use the traditional Caddy's approach. You see here, we could reduce the error by a factor of 10. Results. All right, so all the images are space-time resolved, so we need a fast imaging technique, and here we study the sort of motions, flows, cardiac contractions in the body, and study the effects of the magnetic fields, the spin, so-called spin processing. Here important for study magnetisms. So it's always we do space-time resolved for the imaging. I'm not going to get into details, but we are looking into all kinds of smart strategies. So from so-called compressed sensing, and again deep learning, all that stuff to speed up the. Okay. Now, I am going to show you the example of PhD thesis from the lab so that you can see how we approach the thesis work. This year we graduated actually 3 PhDs, right? So here, Yan Wen, Zhang Hongchu, and Robin Zaffi will show the work very beautifully. And then this is all technology development, but we also are going to show you examples where since originally did more biology work. All right. So Ian has done a lot of work, and this is just a small part of his thesis. Is he look at important work. Look at the signal model. You have the signal magnetic decay, and then phase factor, and then you estimate the fields from the signal, right? And then you do deconvolution. So Yang has is a man of rigor and precision, and you see, you realize these problems with noise modeling in the current approach. So to be more faithful to the data statistics, you need to work with complex signals directly in this manner. And this really brought us a step closer to the truth. Effective, you see, his work won the Best of All prize in QSM reconstruction challenge last year. And you see, it really reduces artifacts. And here is showing examples in vivo imaging. Here we have the hemorrhage in this patient brain, and Yan's work really clean up a lot of artifacts. Chang's work is a step further. Basically, in order to estimate the oxyheme, which related oxygen extraction fractions, so he introduced this model that the oxyhemes are in the cylinders, right? It's a different size, different orientations. They will contribute to a different effect on the signal magnitude, right? So I have an additional term in addition to the signal decay. And then you can model the susceptibility decomposed into the non-blood susceptibility, and then blood susceptibility, which is related, has a term where related oxygen extraction fraction, or the oxyheme stuff. Right, so you propose the solve inverse problems, and then you can determine the oxygen ejection fraction and the so-called non-blood susceptibility, which is something like treating like tissue iron. So we see here in this patient, wake up with a stroke, and we don't know what's going on. Traditional methods, T2 sort of T2 flare and TWI. You see the two lesions there, white and red arrows, but the red arrow has diminished oxygen ejection fraction, meaning that irreversibly that you should not treat those patients because there's a huge risk of hemorrhagic transformation. While the white arrow point to a lesion that is metabolic active can be salvaged. So this is a very, very important information in clinical practice. Right, so Ramin's work is dealing with the fatty tissue, so he needs a model in fat-water difference. Here is the model that the fat has different phase processing because the inaction clouds or the shielding make the fields different. Then you have the additional phase factor there. So to determine all this is so-called to fit data with all these parameters. It's a non-convex problem that it's not a nice problem because it depends on the initialization. To address this problem, he looked into deep learning. So deep neural network, he tried different trainings and supervised, unsupervised, and he discovered this method called no training. So basically, in deep neural network, in the back propagation, you have stochastic gradient descent. So that is sort of like, in fact, different but very effective numerical optimization approach. So you see that this kind of approach and worked and solved this initialization problems. Here he used this UNet, so this is very nice. And okay, so jump into the Synthesis work, which is dealing with biology. So you have this macrophage microglia, a very important part of the immune system. You have the range of the spectrum of activation, so M2 that repairs tissue, and M1 that is pro-inflammatory. It has lots of ions there, and so we can imagine this pro-inflammatory microglia or microglia in the brain. Great. So a disease called multiple sclerosis is a neuroinflammatory disease, and here you see these lesions in this MS brain tissue block, and you see this blood rim. This blood rim, we did elementary analysis. See the iron. It's actually iron. It's actually in this microglia activated, as you see on the CD68 staining. And this provides a very important connections between the MRI, which is macroscopic imaging, with cellular interpretation here. It's actually activated microglia in the brain. And you see in vivo in the patients, this patient has lots of this information going on there, and this provides guidance how to treat those patients. This is very important work. Lastly is the environment. So your PhD thesis work should be in a great environment, which we have the latest and greatest equipment, 70 MR PET, allow you to look at molecular, all kinds of molecular activities, and we have AMR guided focus ultrasound, which allows you to deliver precisely localized energies for therapy and for like brain stimulations. So we have great. So you can also look at the posters outside of Hall 135, which summarizes some of the PhD theses. If you're interested, you can take my class, BME 618, Principles of Magnetic Resonance Imaging. Thank you. Hope this is helpful for you.